Hello, and welcome to this Technology Networks webinar, Integrating Multiomics Analysis in Cancer and Rare Disease Research. I'm Dr. Isabel Ely, Science Writer for Technology Networks and your host for today. We have a fantastic presentation in store for you. Before we dive into the presentation, let me introduce our speakers. Mohammed is a neurogeneticist who primarily focuses on brain-related rare genetic diseases and cancer. He is the founder of GenomeArc and the founding director of the Center for Applied and Translational Genomics at Mohammed bin Rashid University. He is currently working on developing an ASO drug for Dravet syndrome, modeling glioblastoma using advanced machine learning algorithms, and exploring applications of quantum computing.
Hello, everyone, and welcome. I'm Mohammed, founder of GenomeArc, and I'm excited to present our work on building the AI-powered unified multimodal platform for scalable clinical genomics. So we are now entering the era of population scale genomics. Initiative like these, always wonder how we can analyze such a big data set in a reasonable time. So the UK Biobank, if you see, it's actually touching 500,000 samples now. The 470,000 exomes are available. Broad institutes are, almost exponentially sequencing samples. There are also Asia that are catching up with thousands of genomes. So almost all countries have their own population scale genomic projects. This scale demands platforms that are both powerful and flexible enough to manage, interpret, and integrate massive volume of genomic data. Although clinical adoption is slow, clinics and hospitals are integrating clinical genomics unit within their healthcare pathway. More newborn screening programs are announced over the years in different continents. And slowly it's getting a first-year test for rare disease. If you look at rare disease, genomics is extremely effective. Rare disease affect millions worldwide. Most rare diseases are genetic in nature. And we actually saw in OMIM, there are more than 5,000 rare diseases being recorded and documented. Children are disproportionately affected, and the typical diagnostic journey takes up to 5 years, which is also known as diagnostic odyssey. So we believe integrating this multimodal data will able to decipher those complex cases in rare disease that are currently not being diagnosed properly, or at least in timely manner, because the time is a big factor in therapeutic intervention as well. In oncology, the complexity is even greater. We are dealing with germline variants, somatic mutations, and liquid biopsy signals from ctDNA. The molecular insight of these tests are now analyzed together to guide both diagnostics and treatments. So we need a platform that supports this integrated view of oncology, because germline only constitute about 10 to 15% of the diagnostics yield overall in different cancer types. Somatic variants is very complicated to detect small clones, but you have to sequence with higher depth. And liquid biopsy still requires, there needs to be a tremendous effort in algorithmic development to identify what kind of cancer you can predict from ctDNA signals. And the interesting development of clinical genetics is that we are no longer working in genome data alone, because it's kind of limiting if you are only using genomic insight for diagnostic and therapeutics, because there are clinical symptoms data from patients, multi-omics, single cell transcriptomics, and proteomics data. All of these molecular and clinical multimodal data needs to be integrated in a comprehensive and cohesive manner. And if we cannot integrate these multimodal data, we will not have a complete understanding of disease mechanisms, will also impact diagnostics and therapeutics. So one of the biggest challenge in this, large-scale clinical genomic era is not just sequencing, because sequencing is now pretty streamlined. It is actually the interpretation that is the biggest bottleneck for us. Even for some of the known cases, just to process the annotation, it takes hours and hours. So analyzing an entire genome with clinical relevance when it is complex, because you're dealing with millions of variants, small and large, we need a system that go, beyond that timeline and return something very fast. So the diagnostic and intervention decision can happen in minutes, not in hours or days. So we here at GenomeArc built a solution. We call it GenomeArc Horizon, a multimodal platform for clinical diagnostics. It helps in many ways. So our platform offers ultra-fast analysis. Exome interpretation happens within 5 minutes, whole genome within 10 to 15 minutes. It works both locally or in the cloud, ensuring speed, scalability, and compliance with privacy protocols. It's encrypted. It's heavily customizable based on the need of the clinic or labs, and it's technology agnostic. It supports different sequencing technology. So Horizon actually integrates proprietary database, multi-omic database. It connects with the patient health record if it needed to, and there's intuitive interface that is connected with our own AI agent that built within the integrating these data sets. So it is a unified multimodal platform built specifically for clinical genomics. At the heart of this platform is Horizon Intelligence. It is a intelligence that we built to explain all the different features. So Singularity is a feature that we built for rare disease diagnostics. It can analyze a single genome or a trio or a duo for carrier screening. Spectra module been developed for oncology solution, can do genome analysis for constitutive variant findings. Also, it can profile tumors, and also it can predict ctDNA signals. And the last module that we have is the SICRX, is the pharmacogenomics module that predicts the pharmacogenetic variants and also provides a range of drugs options if there is for different gene variants. And if you look at the clinical insight that Horizon provides, it's basically targeted towards rare disease and oncology. We use ACMG guided diagnostics protocol, and that is ultra fast carrier screening for true premarital carrier screening or trio analysis, secondary findings. Tumor profiling is another important task that we do. Tumor somatic variant detection is an automated process when you upload the VCF. Liquid biopsy or ctDNA data can be uploaded to identify the ctDNA variants and its effect and what kind of cancer it might, it predicts what kind of cancer might manifest. And there are a few other modules around oncology, which is implemented in Spectra. And then all these clinical features actually integrates the pharmacogenetic information. You can upload single cell transcriptome data from the tumor or proteomic signatures that will all be integrated into one intuitive platform. I want to share our experience on integrating Horizon with Oxford Nanopore technology using Oxford Nanopore's A100 GPU towers. What we observed that when we took the VCF as input from a long-read genome that constitutes about 5 million small variants, SNVR indels, and about 40 to 50,000 structural variants, it can annotate and predict the variant pathogenicity in 12 minutes, the whole entire genome. And it includes full analysis of the annotation, the variant classification, and also report generation. So all is in one cohesive way. So, and also when you are analyzing the whole genome, you are also in parallel can sequence the whole genome. You can do a LSK whole run, a whole genome run in your P24 or P46 machine, and still can use Horizon to analyze the already sequenced VCF files. So this is an example where we have a rare disease patient using singularity module within Horizon. We identified de novo variants. So because the de novo variants, if you upload trio data, probe and father and mother's VCF file, it can identify automatically all the de novo variants. So this is an example where we identified KMT2D variants that is a known causal variants for neurodevelopmental disorders. And this kind of rapid insight actually can accelerate diagnostics dramatically and also can help a clinician to act in a timely manner for therapeutic intervention. And you can see in the IGV shows that the variant is present in the proband, which is absent in the father and mother. And we have another case involved a colorectal cancer patient. So traditional targeted genetic test failed to explain the cancer or why the toxic reaction was manifested after introducing chemotherapy into this patient. And our pharmacogenomic module from Horizon identified a DPYD deletion variant that is actually linked to the drug metabolism and known to have toxic reaction if the gene is mutant, or the patient is mutant for this gene. And we also found at the same time the cause for the cancer, which was not known. So we found a very interesting mutation in TCF4 gene. So these are examples where you can actually bring different kind of modules in one place and kind of interpret the data, because the clinical data helped us identify the variant that we are looking for in this particular patient. We'll show you how a clinical symptom can be used to generate gene panel, and that can be used in Horizon and link with the patient genome. So our tumor profiling, so the last case we just discussed was for germline variant, but GenomeArc Horizon also equipped with tumor profiling. So in our tumor profiling module, Spectra, we invested a significant amount of algorithmic development. So our platform can detect somatic variants, can classify pathogenicity. It profiles tumor mutation signature to assess the DNA damage. It constructs a full clonal tree for somatic variants, and like how many somatic variants in what clonal complexity. It can identify the tree-like structure for different clones and subclones. And it also can predict from ctDNA what is the cancer that might manifest, because ctDNA will be one of the biggest tests in future for cancer prevention. When you do super deep sequencing of a few targeted genes, whatever the variant you find, you might always struggle to identify the cancer type that might manifest with this particular variant. So in one of these use cases, we uploaded 53 kind of data from ctDNA, and we saw that because it's a pan-cancer gene, and we saw that particular variant have a 43% probability for lung adenocarcinoma, and the probability of other cancer types are also provided. So these are the features that actually comes just from the VCF files that you upload. And it's not just that. Genomics can be very interesting in terms of population-specific variants because from the out of Africa hypothesis, each population actually have some bottlenecks, some rare variants they lost, or some rare variants become common. Even founder effect can be detected. So here's an example where we uploaded a custom database. And you can see on the diagram on your left that all the different ethnicities from Nomad are, there is no bar, there is no frequency of the variant, but the custom database at the end shows it is a common variant in Middle East, actually. So there are a lot of these type of common population-specific variants across the globe, and you can upload your population data into this platform, and it will integrate that population frequency during the pathogenicity calculation. So a lot of our modules have been validated from different sources, and we also looked at very complex cohort of rare disease and cancer cohorts. So this is one of the rare disease cohort where we saw that there is a yield of 36% using our software using Horizon. And it identifies all the known variants and many novel variants. And we also kind of did a big test on the effectiveness and sensitivity of the structural variation pathogenicity prediction for rare disease and inherited cancer. And our ROC analysis shows overall accuracy of 96% in predicting structural variation pathogenicity. So here's a video that I want to show how we use the unstructured clinical phenotype data and predict how we predict disease and create a gene panel. So this is the agent we created using the large language model. So for example, you can use this panel and you can paste or dump any unstructured data. It can come from your health record. It should have the symptoms of the patient. And once you paste the symptom symptom data into the panel, you can update the page with this symptom. What it does, it will bring out all the HPO terms that is relevant to the disease. For this particular example, it is a Duchenne muscular dystrophy description we added, and it identified exactly that particular disease with highest degree of probability, and the disease, and the disease gene it will identify that is associated or known. Not just that, it actually creates a gene panel. So all these genes that you see have different degree of probability. So I want to show you another AI agent that is built within the system. So it's within Horizon Intelligence. Apart from that phenogen module, the phenotype genotype module, you can actually search gene variant. So you can search gene names and ask for a function of a gene that is within your genome. And it will print all the description related to that gene in a very fast manner. So for example, if you have a gene that you don't know what it does, because lots of VUS variants are impacting genes with function that we don't know. So this agent actually can identify all the relevant disease information. Not just that, so if you upload genome within our platform, the VCF files, it learns those variants, and it can find variants within these genomes by typing the variant location, just like that. So it should identify if there's any pathogenic or clinically relevant variants in this location of the genome for any of the patient. So, and what are those patient? What is the phenotype? What is the gene within this impacting this variant? It should identify all these in a small table. So having this option actually helps you analyzing. So this is a frameshift insertion that's been tagged as pathogenic. In many other projects and individuals within this platform that you uploaded. So these are individuals that you have uploaded. So the language model learns those and identified. And if you want to know, so this also identified what gene it is. And if you want to know more about this gene, you can just follow up the question within the language model. So you can see how we are using clinical information and genome at the same time to bring the diagnostic yield. And this actually help us, you know, shorten the diagnostic odyssey time. So in summary, what GenomeArc Horizon actually does, it automates the whole multimodal process. It uses tons of proprietary databases, and it integrates different modalities into one cohesive platform. The algorithms that we use are validated and secure. The large language models comes with many different agents, and we are constantly building different agents for different types of algorithm. And it's ultra fast. It's super fast. It's super secure. It's encrypted, and the way it integrates all these modalities, it will enable early intervention for rare disease and cancer prevention. So this is a disclaimer for Oxford Nanopore technologies. These are for research use only. And thank you. So please let me know if you have any question or concern about the Horizon